Transcription replication conflicts, or TRCs, are a major source of genomic instability in cancer cells. These conflicts occur when DNA replication and transcription machinery interfere with each other, causing replication stress and DNA damage. While harmful in normal biology, this vulnerability is exploitable in cancer cells, which replicate and transcribe DNA at abnormally high rates. One of the central proteins in these processes is pCNA, proliferating cell nuclear antigen. It acts as a sliding clamp, essential for DNA replication, repair, and chromatin remodeling. A cancer-associated isoform, known as capCNA, differs structurally from the form found in normal cells. This distinction offers a therapeutic window to target cancer cells selectively. AOH 1996 is a small molecule designed to exploit this very mechanism. It was developed using crystallography-guided drug design as an improved analog of a previous compound, AOH 1160. AOH 1996 demonstrates enhanced stability, solubility, and oral bioavailability. In preclinical models, it maintains a half-life of over 4 hours in mice and achieves good plasma exposure with oral dosing. Mechanistically, AOH 1996 binds to pCNA and enhances its interaction with RNA polymerase II, particularly its largest subunit, RPB1. This disrupts transcription during DNA synthesis, particularly in the S phase. The resulting interference causes replication fork collapse, leading to transcription-dependent double-strand DNA breaks, and ultimately, apoptosis. This effect is highly selective for cancer cells. In studies involving over 70 different cancer cell lines, AOH 1996 showed broad anti-tumor activity, with a median GI50 of approximately 300 nanomolar. Non-malignant cells were at least 30 times less sensitive. Cell cycle analysis revealed G2/M arrest and an increase in sub-G1 populations, confirming induction of apoptosis. Tunal assays demonstrated clear DNA fragmentation in cancer cells, while sparing normal tissues. When used in combination with chemotherapy agents such as cisplatin and topotecan, AOH 1996 significantly enhanced their cytotoxic effects. These results support its use as part of a combination strategy to improve efficacy or reduce chemotherapy dosing. However, resistance can occur. Mutations in RPB1 can prevent the interaction between RNA polymerase II and pCNA, reducing DNA damage induction and allowing cells to survive. Understanding these resistance pathways is critical for future clinical use. The broader therapeutic strategy here centers on transcription replication conflicts. Cancer cells experience high levels of replication stress due to their rapid division and elevated transcriptional activity. By targeting TRC resolution, AOH 1996 selectively pushes cancer cells beyond their tolerable threshold of genomic stress. This non-oncogene addiction, where cancer cells depend on pathways not directly related to oncogenic mutations, may allow us to overcome resistance to conventional targeted therapies and reduce off-target effects on healthy tissue. While preclinical models, including in vivo studies, show encouraging results, human validation is still needed. Clinical trials will determine the true safety profile, optimal dosing regimens, and the role of AOH 1996 in combination therapies. Investigating its full molecular mechanism may also allow for predictive biomarkers and patient-specific applications. In summary, AOH 1996 represents a novel and promising approach to cancer treatment. By targeting transcription replication conflicts through selective binding to cancer-specific pCNA, it induces DNA damage and apoptosis with minimal toxicity to normal cells. Its synergy with existing chemotherapies and potential to bypass common resistance mechanisms make it a strong candidate for future clinical use. Thank you for watching.